in both wooded and urban areas all over North America. From late March to early May. There are two color variations of the common violet. The picture on the left is the one that you see everywhere with the violet color, and the picture on the right is the second color variation, which is commonly called the Confederate violet. You can see it's white petals with blue radiating from the center. It is very important to study plants because they're essential to all life on Earth. Couldn't really do much without them. So in order to preserve these plants, we must study their pollination because that's how they reproduce. And insects are how flowering plants are pollinated, so we have to study the insects that pollinate them. Our test studies our methods' effectiveness to determine probable pollinators. Our main research focus was to compare the efficiencies among insect pollinators of the genus Viola. We believe that Dipteria and Hymenoptera will be the most efficient pollinators of this genus because of a study that was a similar study done in Europe, which found that Dipteria and Hymenoptera were violet pollinators.
So by testing our hypothesis, we studied two sites that were relatively close together, and both sites contained a large populations of common violet. And we spent 2 days collecting the insects using a variety of ways, and the insects were then the insects being collected were either on the plant or around the study areas. And we preserve the insects by preparing the solutions of 70% ethanol and glycerols in these small containers. And this will help us to preserve both the insects and also to make sure the pollens will stay on them for the later research. We use a gel that was made from a mixture of these materials, and this allowed us to stain the pollens for observations. And this method was from the book that we cite called The Techniques for Pollination Biologists. So we use the gel to swab the insects to get the pollens off them, and then we use the gel to melt it onto a slide for observation under the microscope. So these samples will then compare to a sample of violet pollen, and that will be compared in the same method. So to make sure that our samples was different enough to identify the violet pollen, so we compare the slides to a plant that was the pollen from the plants that was blooming at the same time. So this is a picture of our pollens, violet pollens. And since we're not experts in insect identifications, so we send our specimens to the entomologist and Vanderbilt University for identification.
Okay, our results. We found a total of 55 individuals, 26 from one site and 29 from the other. They belong to a total of 12 different orders. The entomologist was able to identify all of the insects to the level of order, so that was what we used to analyze. That's how we classify them to analyze our data. And we found that 41 of the individuals had pollen on them, and 22 had what was probably violet pollen. So this is just a pie chart of the number of individuals in each order. So this is just the population number. And the orders that had noteworthy numbers of specimen were Araenia, as you'll see. Araenia, which is spiders. Hymenoptera, which is an order that includes bees and wasps, and Diptera, which is flies. So this is a graph of the 12 orders. They're represented there with the population number next to them. Within each order, the yellow bar represents the percent of individuals that had pollen on them. So this is just the percent in each order that were found to have pollen on them. And the green bars represents the percent in each order that had violet pollen on them. And the percents were taken from a total of the order.
To determine the actual pollinators for the common violet, we would need to genetically test each sample of pollen that we got from the insects. And since we didn't have the time to sequence the DNA of each pollen sample, the way that we just talked about, and so our study does not determine the actual violet pollinators, but it points us in the right direction for future research. We considered orders with greater than 50% to be punitive, possible violet pollen to be punitive violet pollen pollinators. Excuse me. Spiders are the most possible pollen, or the most probable pollinators, and we were able to determine this because they had a noteworthy sample size of 9 insects, and they also had greater than 50% possible violet pollen. Despite what we had previously believed, Diptera and Hymenoptera were not probable violet pollinators, even though they had 17 and 15 specimens respectively. They had less than 50% possible violet pollen on them. Since the study was to test the methods of effectiveness to determine pollinators, the next step would be to use the study to test other plant species. It would be very useful to use the study on endangered plants to help further preserve them. And also, it would be good to collect the same, at least a similar number of specimens in each order size so that it is not a limiting factor like it became in our study.
So finally, we would like to thank Denise Ware and Christine Weiser from the Sharpie Bottom Natural Center for all the sources, and Denise Dugu-Wings and Daniel Francis Lab from Vanderbilt University for the help of methods, and our instructors from School of Science Math, Dr. Bonnet, Dr. Creamer, Dr. Eads, and Dr. McHugh for all their help and support. And finally, again, the Tennessee Junior Academy for Science for giving us an opportunity to present today.
You had mentioned spiders. That's the 8-leg arachnid. So they're probably looking for lunch.
We actually found a lot of spiders that were living on the flowers, which would be why they had violet pollen, but most pollination is just from contact. So if an insect had pollen on it and it walked on the flower, on a different flower, it could pollinate it. And so spiders really aren't normally thought of as pollinators, but the fact that they live in such close contact with these flowers means that they might actually be pollinators.
Because the pollen needs legs or wings to go somewhere to be able to pollinate, so it uses spiders and other insects.
Now that you know how difficult it is to make a definite conclusion of pollinators, do you think the European study do you believe their results, or do you think they
Yes, they actually did a much more extensive study. It was several years ago, but I think it was actually in the 70s. But they were able to do some genetic testing, and they actually were able to observe how the flowers were pollinated. So how flies in particular, how they went into the flower and how that would release the pollen in the flower. So it was a much more extensive study, and that would be a way to find out what the actual pollinators were. But this is just a good base that we know
years on their study and has lots more resources than we did.